All right. So, hello and welcome to today's episode of the Tech Transfer Edge, where we discuss operational challenges and technology transfer in cell and gene therapy. Today, I'm joined with Lee Buckler. Lee, how are you?
I'm well, Nicholas. Thanks for having me. Good chatting.
Awesome. Well, I'm excited to have you on the show today, Lee. So previously, we sort of have discussed many of the challenges in translating sort of research to manufacturing in the cell and gene therapy space. And one thing that caught my attention with one of our previous discussion was about the sort of widget theory. You kind of shared that, well, products are unique. A lot of the manufacturing steps, like the unit operations, are identical. And companies sometimes act like everything is bespoke because of IP lawyers. I'd love to sort of just hear have you share your thoughts a little bit on that and why that might be the case.
Yeah, thanks, Nicholas. Great question. And it's a complex answer, of course. As you suspect. I think if you think about any sector that goes from being highly innovative, research based, through all the way to the other end of the spectrum where it's highly commoditized and standardized and uniform, and we're squeezing, Six Sigma sort of, Lean Six Sigma compliance out of every step, and margins are small. We're still way over on the left hand side of this continuum, right? We're still just emerging. We now have several commercial products. And we're starting to see where the similarities are, but even those are evolving very rapidly, right? I mean, if you look even within, Novartis was the first one to commercialize a CAR T cell therapy, and now they're working already on 2.0 or 3.0 versions of CAR Ts, where the manufacturing of even a CD19 CAR T looks radically different than it did 7 years ago. And so, we're still very rapidly evolving. And so, it's difficult in that kind of environment to race to standardizing too early because you're standardizing against something that's still rapidly evolving. And so I think there's this tension between wanting to standardize, wanting to create common, widgets where there are common sort of processes. But if you lock in that too early, you depress the need or the advantages of the innovation that's still happening, right? So so we see certain things which are highly uniform. Many people are using the G-Rex as a common tool. Many people are using Lenti as a viral transfection tool. Many people are using certain kinds of bioreactors, certain kind of vectors, certain kinds of reagents. And yet we're also seeing now a wave of other kinds of bioreactors, other kinds of vectors, non-vector sort of ways of getting things into cells. And so there's a lot of really exciting tension between wanting to squeeze margins and commoditize and standardize versus wanting to still be highly innovative. And that's the tension we're very much in right now, right? And so I think, where that leads us to is generally a resistance to want to include too many of the steps in a single box.
Okay.
Because the level of innovation that's happening in one of the processes in a multi-process manufacturing paradigm might be happening faster than another step. And so if you've locked all of your steps into one box, it doesn't allow you to innovate maybe as much as you would like on one of those steps faster or more than you might want to be prepared to innovate for in another step, right? And so I think we're, although there are some great people working on multi-step boxes that I love and adore, and I think there's some advantages to those clearly. I think many are still preferring to standardize and automate each step in their preferred box or preferred widget, and then try and find ways to close and automate and digitize them together in a daisy chain of technologies. at this stage, given how far left we are in on the innovation continuum.
Okay, that makes a lot of sense. So it sounds sort of like you need a certain level of flexibility at the current stage of development where the technologies are right now. You need some flexibility as to sort of the edges to them. It's not like an electrical socket where you can just plug and play everything together. And in order to really get the effect you want, you need to have that level of flexibility as to the exact edges and how you're going to connect them.
Yeah. Now, the tensions then starts to be very contextual, right? So, if I'm producing, if I want to produce thousands, maybe aspirationally tens of thousands of doses of a single product, and I know that my BLA, that the terms of my CMC condition are going to be very difficult to change, although it's always subject to changeability. If you can prove comparability, you can always change your manufacturing. So as long as you can show comparability, you can change manufacturing even post approval. But if you really feel like you've locked in your manufacturing for this particular product,
Mhm.
then you might be more um, 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 aligned with wanting to have everything in a box and just shrink your factory down to as small as possible and produce as many of those doses in as small a clean room footprint as possible. Because where we're still not today, even though we have systems which are closed, and we have systems which are highly automated, and systems which allege to be clean rooms in a box,
Mhm.
we're not yet in the regulatory environment. We haven't seen yet a massive move to being allowed to operate those boxes outside of highly controlled clean rooms that we're used to even doing open manual processes in, right?
Mhm.
So, one can think aspirationally that that's where we're pointed to in the future, because we manufacture many biologics today in very uncontrolled rooms, or at least very, very low controlled environments. Um, but we're not there yet, right? And so we still are operating. Even if you were to get to the point where you were manufacturing in a single box all of your processes, um, we still have to operate in the clean room. So right now, it's not about getting out of the clean room. It's about operating as much efficiency and manufacturing as many doses in a single clean room as possible. Um, is the ROI there, right? And then then the other question becomes, okay, do I want to spend what I need to spend on those multi-process boxes to manufacture this product, when I know that version 2 of the product is already in my clinical pipeline, and the manufacturing of that is going to be radically different, right? And that becomes an ROI thing, for the sponsors to measure. And keep in mind, we're still not at the point where any one of these companies is producing, you know, tens of thousands of doses per year for a product, right? I mean, we're
Mhm.
we're we're barely producing that many per year in aggregate of all the cell therapies, being produced. So, there's a long way to go in terms of the kinds of scale that would allow us to think differently about the ROI, for these things than we are today.
Okay, so the first thing that comes to mind when you're kind of talking about locking in these processes and about this sort of tension between innovation and manufacturing is in the context of technology transfer. So ideally, surely all the innovation should happen at the very beginning, and you should figure out your process and then say, okay, great, I've got it completely characterized. Now I'm going to go to manufacture. Is this a reality? And like, how does that actually play out?
Yeah, I mean, I think that the question becomes when you're right, when you turn it onto your manufacturing line, then your goal becomes something very different. You're no longer
Mhm.
wanting to innovate. You don't want innovation anymore. You want to manufacture consistently, reliably the same product every single dose. And of course, that's a different metric. And there the goal becomes, can we eliminate biological differences,
Mhm.
or can we compensate for biological differences?
Mhm.
And can we also eliminate the potential for human error.
Yes.
So to the extent that we have open manual processes that require human discretion, human evaluation, human analytics, and human processing,
Mm-hmm.
we're subject to human error. The more we can lock that up, we can digitize analytics, we can, machine learn, we can close processes so that the human is just pushing a button instead of, pipetting and processing and selecting. Then we can start to eliminate human error and move toward more and more consistent, reliable production. Now, in the context of autologous cell therapies, of course, that becomes eminently more difficult because we have more of the former problem. We have the biological variability of the input, right? The input material is biologically different every single time. And so today we're ironically still, from a manufacturing perspective, if you come from this to this discipline from another sector, you would be shocked and horrified that many of our products today are being released and clinically infused out of spec. Right?
So maybe to clarify, just to start with the autologous, are we talking about differences between patients? Is that the variability you're describing?
Yeah, because the autologous, by its very nature, the starting material of the manufactured product starts from the patient. So we know that biologically, you and I are different out of the gate, even if we were both healthy, the same age, even the same parents, biologically, we're invariably quite distinct. But then you factor in the fact that we've got disease now, and we've been, our body's been beat up by, 2, 3, 4 standard of care treatments. And so when we go to harvest your immune system cells to engineer them into a therapy, we find someone who not only is biologically different than the last patient, but is biologically destroyed, right? Because of all of the disease, the ravages of the disease, and the ravages of the treatments they've had before we got to them.
Yeah. We're talking about cancer patients a lot of the time.
Yeah, exactly. Most of these cell therapies are still for cancer patients. They started out being initially approved as last line treatments. So these are patients who are otherwise in 4 weeks going to die, right? So their bodies are in a horrible condition. Now, as we move toward, second, first line treatments, which we are now moving toward, and have achieved some, the variability due to disease is less, but we still have the inherent biological differences of humanity. And so the question becomes, do you define the specifications of your product with enough latitude to compensate for that? Probably we do some of that, right? Also, do we have enough in-process analytics that we can make sure that, when one patient's cells takes 14 days to manufacture and another patient's cells only take 8, that we have enough analytics to know that we're going to need 14 for the first patient, but we only need 8, and so we can cut off manufacturing at 8 days instead of manufacturing everybody's for 14 days, right? If we can gain, if you can gain every day of efficiency you can gain is huge dollars, right, in terms of cost of goods. And so, can we build in enough in-process analytics for us to be able to make the human decision to cut it off at day 8, right? Or can we embed enough AI or machine learning so that the machine can tell us, okay, you've achieved your manufacturing specifications at day 8, you can now harvest, right? We started, less than 10 years ago with the former, right? Everybody's product was manufactured 14 days, whether it needed to or not. We didn't have in-process analytics. We started to move toward having just enough insight to be able to make human decisions. And now we're starting to see examples of machine learning where we've created ecosystems of data, feedback loops, where the machines can learn to assess and inform us whether or not it believes that it has achieved specifications at what point early, and this goes not only to meeting specifications, but it also goes to things like media consumption. So, media the media to feed these cells during expansion is one of the most expensive components of manufacturing.
Mhm.
So if we can be evaluating the health of the cells in the bioreactor and understand, okay, I don't need to exchange as much media as I thought I did, or I just need to add a little bit of this additive or less additive than I thought I did. Now I can really start to have more and more control, not only of the product meeting specifications, but of the cost of producing that product. Because now I'm really only adjusting media consumption based on what I'm assessing in real time.
Yeah, that makes a lot of sense. It sounds like generally the more control, more characterization, and consequently control you have over the process, the more, well, the less waste there is in general. And in very expensive processes, there's a lot of return there.
Right. Right. And that's where, where we're starting to see a lot of these closed systems. They're not just closed or not just automated. They're also getting smart.
Mhm.
Right. For that, you need not only biosensors, ways to analyze in process what's happening based on what do you think you need to be measuring, but then also ideally to be feeding that back into the machine to be learning over time, right? Or at least, or into an outside machine model, right? Where you're feeding data into a model so that you can learn over time, right? And that's the advantage of these systems right now. I think it's not the ROI at the moment is to feed into manufacturing innovations that can optimize the cost of goods and optimize the product meeting specifications, more so than necessarily squeezing margins and getting manufacturing efficiencies per se.
That makes sense. You mentioned sort of that some of these autologous therapies, they get released out of spec, and that would surprise someone from outside the industry. I'd love to hear a bit about that.
So, keep in mind that most of these therapies, particularly last line therapies, these patients have run out of options.
Yeah.
So from a clinician's perspective, the doctors have a great deal of latitude to be able to use whatever treatment they believe might be in the patient's best interest, that's safe to do in the context of that patient's disease progression and that doctor's discretion. And so, that's why we have off-label use of drugs, right? Because we've given clinicians the latitude to use their medical discretion to treat patients as they deem fit, even though the product has not been labeled, as such. And so a corollary to that is that physicians have the ability to be able to decide that a cell therapy, even out of spec, is the client's best option.
Okay.
And so if you've got a client on their deathbed and they've run out of standard of care options and the CAR T, is slightly short or maybe even a lot short of the cells that you think it needs, or but you think, look, we know at least from the safety profile of these cells that it's not going to do any harm.
Hmm.
And if we infuse the patient with this out of spec CAR T, it has the potential to maybe do some good. Why not do it, right? That's the clinical discretion that doctors have. And so, and so that's one example of out of spec analytics. And then you have a product which meets specs, but a lot of times we don't have the full release panel yet. So if you
Hmm.
look at sterility testing, for instance, a lot of times you don't get your full sterility panel till 14 days after manufacturing release, because
Right.
they're based on culture methods,
Yeah.
right?
Yes.
And so in almost every instance, we're infusing the product, trusting that it's sterile, even though we don't have sterility data.
And okay, something that's sort of come up, it's talking just about this with some previous guests, was about sort of given the risks and where these patients are at when they're getting infused, that they're sort of infused at risk in a sense for the sterility testing, in that,
Right.
if it's microbial sterility that you're worried about, you have antibiotics on hand, and you're
Right.
just prepared for those to be there.
Right, because so there's two things, right? There's the out of spec product being infused, and then there's at risk product, right? Which is the two proper labels for the things I just described. And
Okay.
you're right, because at that point the doctor's discretion is, look, We know that it's, the sponsors told us it was manufactured, per protocol. There was no deviance in terms of manufacturing. CMC's all been checked off. QA has signed off on this. But as a matter of routine practice, we can't prove sterility until it's too late, you know? And so we want to infuse these patients. And so you infuse them at risk, and then you get the sterility. Now, what's happening is we are innovating around those problems where we will have much faster analytics. But at the moment, the analytics technologies are way behind our ability to manufacture these technology. Most of the technology innovation has occurred around the manufacturing, and maybe to some degree a little bit less on the vectors used. That's catching up, and the analytics has been the very poor, third removed second cousin of all of this. It just has, and I think it's because, there's a whole bunch of reasons, but these therapies were approved very quickly, and so the technology sector is playing catch up. And so you can. Some people love to blame the regulators on this. The regulators are being naturally risk averse in terms of accepting surrogate analytical technologies, release technologies. But I think there's a very active innovation and regulatory discussion happening to allow us to be able to bring on new analytic technologies to be able to release these things, not at risk, but we need non-culture methods, for example, for sterility. We need fast PCR. And again, even on the analytics, we also want to remove the ability, the susceptibility to either human error or human discretion, right? So we don't want somebody looking at the cells and saying, oh, they look good, and that's our QC test, right? Which in the early days of stem cell transplant, that was So we want them push button, we want them fast, we want them controlled, we want them small, we want them, and so the analytics is behind, but there's a lot being invested now in that.
So something that sort of brings to mind is it seems, and I don't know if it's just a characteristic of being a new field and technology overall, but it seems like there's a lot of these sort of judgment calls. If we compare it to, as an example, like auto manufacturing, completely standardized, happens in a factory. No one really needs to understand to that extent. Most people don't have to understand to that extent how the whole thing works. It seems as though, for example, like a scientist looking under a microscope, they might say like, well, these cells look happy. I'll sign off on them. And I'm curious to hear your perspective on this. Do you see that there is a lot of judgment in this sort of space?
I think we've moved beyond cells look happy as a QC release, but so there's almost always some cell counting, VCN, sterility, mycoplasma, potency test that's applied, that's data driven. Where the discretion comes at the moment is either the at risk or the out of spec, but that's more of a clinical decision and regulatory decision. But I think where we're more susceptible is around the reliability of the specifications that are set in the early days. So and the technologies used. For example, cell counts. There's lots of different ways to count cells, and a lot of cell counts actually are highly susceptible to counting dead cells, right?
Yes.
So you're counting something, but are you counting what matters, right?
Yes. Yeah. Exactly.
That's the real question, right? And so I think you can say it's data driven, and there is data, but is the data really speaking to what the data is supposed to be evaluating?
Right.
And I think that's the real question where I think the innovation is happening. And so it's less about human discretion at the manufacturing QC stage as it is
Mhm.
about the who's setting these specifications and what technologies are being employed and selected, to evaluate the specs, right? So A, what specs are used, B, what technologies are being used to provide the data to demonstrate compliance with the specs. I think there's a lot of room for improvement there.
Okay, that's sort of what I'm curious about. And so those sort of standards, like along, if we think about the sort of innovation life cycle of a medicine, when do you have to decide those?
Well, those get, I mean, it's an evolving, paradigm, I suppose. For instance, the FDA commonly says in phase one, you don't need your full release panel set,
Mhm.
but by phase three, you do.
Okay.
So somewhere between phase one and phase three, you've got to finalize your QC the specifications of your CMC and the specifications by which you will release the product. And then prove in phase three that you can do that, and those become locked in for your BLA.
Okay.
Right? Oftentimes developers will measure a whole bunch of things in phase one,
Mhm.
knowing that they're going to have to pick, ranges based on data collected in phase one, ranges, specifications, start to tighten those up in phase two and lock it in phase three.
Okay. That makes sense. So it's sort of a phase wise approach.
Yeah.
I'm curious about, like we were talking earlier about the plug and play technologies and the flexibility that's needed. So would you say that these more standardized technologies that have sort of rigid edges and barriers, are those better suited for the later stages of clinical development?
This is a fascinating debate, right? Because we have seen this. This has been the tradition. The tradition is we start with open manual processes, flasks and open processes in clean rooms, thinking we can automate and scale and worry about cost of goods as we later,
Right.
right? Then that's the typical sort of, I'm a scientist, I'm not a manufacturing person, so I'll just
Mhm.
start my phase one, with a process that I used in the lab, preclinical,
Yep.
because I know it works and I don't have to worry. I'm not only producing 80 doses for or 30 doses or 12 doses for my phase one. So I don't
Yep.
really need to think about scalability, ability, anything, right?
Yep.
Um, and that has been our tradition.
Right.
And I think there are examples of people who have scaled from there and got into something that's relatively scalable. The challenge, of course, is every time you do a significant manufacturing change, you have to prove comparability.
Right.
And um, I was, for instance, in running an autologous clinical cell therapy company. The product was manufactured using a particular cell culture media, and the cell culture media went off the market.
Yep.
And
Supply
when
risk.
we thought, oh, we'll just insert, alternative media B, the cells flatlined,
Okay.
right?
Yeah.
And we didn't know why,
Yeah.
right? That could be a company killer,
Yeah.
right? And so, those are inherent challenges that exist all the time anyway. But when you're suddenly trying to move from a flask to a stir tank on beads or a fixed bed bioreactor or any of other whole fiber, any other kind of thing other than a flask, that's a
Right.
significant manufacturing change, and you've got to be able to prove comparability, and it may or may not work.
Mhm.
And so there are people, I know you've talked to Jason
Uh,
Foster at Ori, who is a huge proponent of start with what you want to end with. What are you, crazy? Like,
yeah.
don't start with something you know is never going to be scalable,
Yes.
right? Like,
Yeah.
why would you do that when
Mhm.
you have the option? Now, in the early 80s, we didn't have any options, right? We didn't have the option to do anything different. But
Yeah.
now, if you have these machines available to you, why wouldn't you use something that it has the inherent ability to scale?
Yeah.
right from day one, because you know you're on a scalable platform,
Yeah.
right? And there's a lot of argument for that. I think there's a lot of reasons to think that. Now, the argument against it is, yes, but my Flask is, 1/100th the cost of your platform, and it's highly expensive consumable set, that you want me to use. I think there's arguments on both ends of that spectrum, and there's lots of middle ground in between those things, right? Some, I don't think Ori is necessarily one of those, but some have created RUO or small scale versions of their platform for people to
Mhm.
use at early stage research and even maybe small clinical trials,
Mhm.
that are cheaper to use. And then you can scale, arguably transfer your manufacturing into the GMP equivalent with less risk than into a completely different platform. So there are some that try to provide that bridge, to ease the pain of being in a scalable platform from day one.
Yeah, and that makes a lot of sense. So one thing I've noticed, like with Ori's platform, is that they one of their values propositions is reducing tech transfer time, and it seems because it's like a one-to-one comparison. Like you've got the same thing you're starting with and transferring into a very similar machine on the other side. So I'm curious, with the current manufacturing processes and CDMOs that might be doing like equivalent manual processes, maybe we'll call them, like, is that just because the scientists at the beginning went with what they were familiar with, and there was sort of no other choice?
In the early days, there was very few choices. Yeah.
Okay.
Right. I mean, you look at Kymriah. Novartis basically commercialized what and how UPenn was manufacturing.
Right.
Right. There was some innovation and optimization, but basically they went commercial with what they bought, right?
Mhm.
And what they were doing at UPenn. And there's probably a lot of really good reason for that, right? A, there wasn't a lot of options. B, there was tons of innovation risk already with trying to commercialize the first CAR T cell therapy without having to also take the risk of a completely different manufacturing platform than Carl and Bruce had used in their early clinical work, right?
Right.
So why take on that additional risk? Let's see if we can actually get this approved and through commercial, and so that's one logical reason for that decision, I'm sure. Now there are more options.
Mhm.
um, there are more options, and and if you are trying to commercialize a Me Too CD19, CAR T, you've got more incentive to try and distinguish what and how you're manufacturing from others, right?
Mhm.
Because I'm producing it exactly the same way, exactly the same thing for exactly the same indications. Doesn't seem very logical, right?
Yeah.
So you've got incentive to also take on manufacturing innovation. I wouldn't say that the ease of tech transfer, from phase to phase is necessarily something that's unique to Ori. If you've got a manufacturing platform, whether any of these manufacturing platforms now that offer similar sort of multi-step manufacturing units, that's the argument in their favor, right? Is you're not you can start with this, you can scale with it.
Yeah. That makes a
I
lot of
think the
sense.
real I think the real distinguishing comes with you know um, how controllable is it,
Mhm.
right? So you definitely want you want to be able to lock it down
Yeah.
so that users can't control things they shouldn't control
Mhm.
once you get to that point. But in the early phase, the advantage of using these open systems is that you can still play with it,
Yeah.
and you can innovate, your manufacturing in the early clinical. Whereas if the box is too locked up
Mhm.
right from day one and you can't really innovate still, the the early phase scientists and process developers won't like that, right?
Yeah.
So in the early days, you like the ability to have a range of controls that you can play with and implement, right, and optimize around. But the ability to lock it down when you need to lock it down, in late stage. And I think combined with that comes, how smart is it? What is it measuring? How much data is it giving me? And can I control for those, and is the machine learning with me? Right. I think that's kind of where we're at in terms of distinguishing one box from another. I think we're seeing some divergence there in terms of what are we measuring, how smart is it, how much data, how much in-process analytics do we have, is there any machine learning in it? And then we're back to the classic conflict between wanting your box to do as many steps as possible, or separate out the steps, because I might not want all the steps done in one box. You could imagine, for instance, if your cell isolation is done in the same box as your expansion takes 3 days, 4 days, 7 days, and isolation takes 2 hours. You've now locked up your ability to do cell isolations in that box until your manufacturing's done.
Yeah.
So if your cell isolation is being done in a different box, you can run 20 cell isolation processes and feed them into different expansion boxes, right?
Yeah.
And so you're not tying up all your unit process in a box that's being underutilized for that step.
Yeah, that makes a lot of sense. You can't really do much in parallel if it's all contained and you can't get in or out.
Right.
Yeah. So one thing I'd be curious to discuss is about the sort of the difference maybe in mentality between innovators and recipe followers. So like, CDMOs traditionally kind of just like a factory. You've got innovative companies doing R&D. With the current stage of affairs, is that sort of like a hard, clean break, or is there some overlap there?
I think there's overlap.
Okay.
And I think there's examples of this even from other biological sectors, right? I mean, if you look at antibodies, I wasn't around or involved in the sector at the time, but as the story goes, Lonza essentially became the go-to contract manufacturer for antibody therapeutics because they innovated a way to produce them logarithmically cheaper than anybody else had.
Right.
And so if you wanted to compete, you had to go to Lonza or find or innovate a competitive technology, which in the early days, hadn't been done. And so Lonza distinguished this, just drove everybody to using them, right? Because they'd innovated. They weren't recipe follows. They had enough customers where they actually invested in an innovation breakthrough that allowed them to produce the therapies much cheaper, and so it drove the customers to them. And maybe that innovation was driven by one customer. I don't know the granularity of the story, but that's the story as it's been passed down to me. And so I think there is opportunity for CDMOs to innovate as a business strategy. And so they can innovate. They can also take bets, right? So right now we're at this stage where we have a whole bunch of different manufacturing boxes out there, right? And you could take a bet on one of those boxes and double down on saying, okay, I'm going to become this company, like a Coke shop versus a Pepsi shop, right? And BMS has done that with Solaris, right? So BMS has said we're going to bet on the Solaris system. We're going to invest in the company, and we're going to put these units in our factories or contract our manufacturing to Solaris. There's that option, right? The other option, which many of the CDMOs, I'll say most of the CDMOs today, don't want to make that bet. They don't want to bet on Coke versus Pepsi. It's too early to know whether
Yeah.
there's winners or whether it's just going to be like Coke and Pepsi, just take your market share, everybody wins, or
Yeah.
whether there's a winners and losers, and they don't want to make that bet. And so they're waiting for the customers to say, okay, we want you to manufacture and we use this box. So if you invest in this box, you bring it into your factory, prove that you can do it, then you'll win the manufacturing business. So most CDMOs today don't want to bet on the tech, on the manufacturing tech. They want the customers to bet on it, and then they'll implement it.
Okay, so
The
that makes
challenge
sense.
the challenge with that, of course, is that especially in a market where we are today, where there is overcapacity, everybody's risk averse and a recipe follower. And so there's not a lot distinguishing between one CDMO and another CDMO. They all have clean rooms, and they're all waiting for somebody to tell them which box to use.
Yep.
Right. And I think we're starting to see some CDMOs, especially in this market where, you're desperate for business. You've got to distinguish yourself from your competitor. The only way to do that is to bet on a box or bet on a tech or innovate. Right. And so you look at Omnia Bio, for instance, in Canada, they're really making a bet on AI, machine learning, that's one of their bets. Every CDMO has had a tendency to develop expertise around certain cell types.
Yep.
So it's like, oh, you go to this, they have a lot of experience in autologous, immune system engineering. This shop has a lot of experience with, adherent cells.
Yeah.
Has a lot of experience with iPSCs. That's just a function of kind of where the business has grown and the kind of expertise they have. But the tech innovation has been one that CDMOs have been reluctant to make. And I think, the challenge with it is it's inherent in their shareholder structure.
How so?
Well, if you think about it, CDMOs are typically factories where you want to maximize, utilization rates of the infrastructure that you've built, and it's a high volume, low margin business,
Right?
And the return is on utilization, right?
Yeah.
Keeping your personnel cost, commodifying and just standardizing everything as much as possible, and getting high volume, high utilization rates, and your margin, because your margins are low, and that's your dependable return, right?
Right.
It's not built. The shareholders haven't thrown enough money at these places to now take technology risk.
Yes.
Right. And so it takes a unique CDMO shareholder base to agree to let them take technology risk, because arguably it's another set of risk, and it probably requires a different capitalization.
That makes sense. And, just thinking from the type of person that would invest in either like an innovative business or a manufacturing one, I'm imagining that's probably generally lower risk takers are the people who are going to invest in a factory, and they probably don't want to take technology risk, or they would be investing in the actual assets.
Yeah. I mean, they're both great ways to make money, but they're two very different risk profiles and very different investors, to your point, right? And, if I can invest in the factory that makes the iPhone, or I can invest in the next, iPhone, killer. That's a very different risk appetite profile and probably capital requirement.
That makes sense. So something I've noticed after speaking to a few CDMOs is that they do have a very wide variety of options in terms of the technologies available. And one question I was sort of wondering, seeing this, was like, wow, that must be operationally really challenging to manage this. Like, why would you have 8 bioreactors? And this sort of explains that when you don't want to be the one who actually makes that call, and it sort of reduces your market exposure. You want your customer to make that call.
Yeah. And I think when you dig deep in it, everybody says, oh yeah, we can use that system, that system, that system, that system. Most of them, if you really looked hard and asked the right question, probably don't have all of those systems in their shop. They're confident they can use it. They're confident if the right customer comes along, they will buy it and use it. They'll figure it out. That's what they're selling. But it would be a very expensive toy shop to have all those toys and no one playing with them, and you're just waiting. It's a field of dreams, a very expensive field of dreams strategy to have every system in your shop, and only half of them are actually being used.
That makes a lot of
Right.
sense.
So it would be, I don't know any CDMOs that have, have doubled down on all the systems, just waiting for the customer to come along to use it. The other interesting approach to this, of course, is what Miltenny's done. So Miltenny has the toolbox, and they now have started a CDMO business built around their toolbox.
Yeah, the
Right?
technology first, and then building the infrastructure around it.
Right, exactly. And so what they say is, look, if you use somebody else's toy, we're not your shop.
Yeah.
But if you use our toy, there's no better CDMO than us, because we know how to optimize this in ways nobody else can even imagine, because we own the machine. We can unlock things nobody else can even unlock, because we own it. We know
Yep.
it. We built it. We designed it. We're constantly innovating around it. And I know, for instance, that a company, for instance, that has that selling proposition has been very, very compelling, because they're like, well, we do use your machine, and so why would we go to any other CDM? Because we were having some challenges around the machine, getting what we want out of it. So if anybody could help us figure out how to get what we want out of this machine, it's going to be them.
Yeah, the level of technical control is, it's unmatched. That makes sense.
And I think that's also what's interesting. Solaris has done a very similar thing. I mean, they started out as a box company, right? We're just going to sell our box. But the box was really big. The box is really expensive, and they thought, rather than trying to support at this stage, selling a box to every hospital, in a decentralized manufacturing strategy, which, you look back at the record, as far as I read it, is what they started out with. They said, no, we're going to create the factory of the future. We're going to put our boxes in our factory, and become a CDMO, of Solaris systems.
That makes sense. Well, as we're sort of wrapping up, sort of one last question I'd love to sort of end with is, looking at the various technologies and the current state of affairs. We've talked about bioreactors, process analytics. What would, if you could snap your fingers and fix one of these problems, which one would you pick?
Well, my favorite problem right now is analytics. Right.
Okay.
I've talked about this gap between, the current analytics that we're using, and the time and the labor and the cost of them, versus what we need, right? So there's this huge opportunity gap to take even existing. We don't even need to necessarily invent new technologies. We just need to prove that existing technologies can be applied in this sector, right?
Yeah.
So there's a lot of overlap, obviously, between diagnostic technologies and analytic technologies, right? The diagnostics companies very rarely know anything about bioproduction. But the same technology that can be used for diagnostics oftentimes can be translated into bioproduction QC applications, in the right hands, right? So I think there's a lot of opportunity there. I think that's my favorite problem right now, and something we're spending a lot of time looking at. I think that's my the short-term problem I want to tackle. The long-term problem that I think, I don't think there's much disagreement. There might be some disagreement as to the pace at which this will happen. I think ultimately, these systems that we're talking about, whether it's a closed daisy chain of systems or a single systems that all the unit processes are done in, like a clean room in a box, like a Solaris or Multiply Labs or Cellular Origins are building, right, where you can put different kinds of machines and robots inside the box, but the box is proven clean. Will drive us toward, and I alluded to this at the beginning of our chat, drive us to lower and lower environments. Ultimately, I believe,
Like
if you look at
lower as in less controlled.
less controlled. Yeah,
Okay.
Ultimately, you look at other biologics production. It's done in closed systems I like to say dirty rooms. Nobody likes me to say dirty rooms, but
Yeah.
but rooms that are a lot less expensive, right?
Yeah.
In terms of the level of air quality, right? And that's because we've proven over some period of time and with enough data that it's impossible to contaminate,
Mhm.
right? What's going on inside the box, and also you're not going to get, people get very exercised about the vectors. Oh my God, what if we release, vector into the air, right? And whether that requires envelopes over them or lower controlled envelopes that in the case of catastrophe will contain viruses, for instance. I'm convinced that ultimately, whether it's by just lowering the controlled environment that we're operating in, or in the case of us not worrying about viruses, non-vector, where we have non-vector applications, just I mean, if you look like at a blood center, for instance, I'm heavily involved in Blood Centers of America. Blood is processed, tested, packaged, stored in low controlled environments every day, and used for clinical transfusion, and then same with many of the other biologics that we use as clinical treatments today, right? So ultimately, I think that's what's going to strip out a lot of the cost of these things. A lot of the cost associated with these therapies is around the clean rooms that we have to operate in, right? Even if we have controlled, automated, using to do this. Now, the other part of this debate, and we won't get into, we don't have time for it today, but is we often talk about the cost of these therapies,
Mhm.
and there is a cost of production which we can and are addressing, but that's highly divorced from the price of the therapies, right?
Yeah.
When you have a curative therapy where you can eliminate the cost of chronic care, or highly improve the quality of life or highly extend life, that allows a price which is quite different. It's highly, when you're looking at the value of a therapeutic,
Yes.
that allows you to price it based on the value of it. That's highly disassociated from cost pricing, right?
Yes. And that's unique from something that you might take weekly.
Exactly. Yeah. And that's not just true of cell therapies, but it's certainly true in cell therapies.
Wow. Super insightful conversation. So yeah, thanks so much for joining us today, Lee.
Yeah, thanks a lot for the insightful questions, Nicholas. It's always good to chat with you.